afternoon, everyone. My name is Beatriz Cantada. I use a she pronoun series. I'm the Director of Engagement for Diversity and Inclusion in the Institute Community and Equity Office. We begin our events with a land acknowledgement. MIT acknowledges Indigenous peoples as the traditional stewards of the land and the enduring relationship that exists between them and their traditional territories. The land on which we sit is the traditional unceded territory of the Wampanoag Nation. We acknowledge the painful history of genocide and forced occupation of their territory, and we honor and respect the many diverse Indigenous people connected to this land on which we gather from time immemorial. Indigenous peoples have always practiced the honoring of guests as well as their hosts when visiting other nations and communities, and that's why we start with a land acknowledgement. So we'd also like to do a few acknowledgements. Thank you to our resident bouncer, DJ Rachel O. Other acknowledgements in this space, we have Tim Jameson, Associate Provost, John Dozier, Institute Community and Equity Officer, our colleagues at Lincoln Lab. I know Ben McDonald's faculty host, Tim Swager, couldn't be here today, but definitely we recognize and appreciate his support of this program and of Ben as well. I also noticed some of our MLK cohort members in this space. I see Charles Sentio, Jamie Macbeth, Luis Gilberto, Murillo Urrutia. Am I missing anyone else? And our speaker this afternoon is Dr. Ben McDonald. Ben is a postdoctoral scholar in the Swager Group at MIT, utilizing synthetic chemistry to develop new functional organic materials for chemical warfare agent responsive surfaces and dynamic biphasic emulsions. He earned his PhD in chemistry from Northwestern University in 2018, where as an NIH pre-doctoral fellow, he explored the total synthesis of polyphenol natural products and the development of new chemical methods for small molecular synthesis. Ben is an aspiring academic who, beyond his scientific work, seeks to use his position of privilege to normalize intellectual inquiry in young students of color. Right before we opened the doors, we were talking about synchronicity. So last year, Ben was the first scholar to shift to a Zoom virtual workshop. And today, he is the last scholar for this academic year to finish up with a Zoom workshop, because we are hoping that come September, we will now shift to a more hybrid environment and maybe see each other in person and share a meal. So again, Ben, we come full circle and appreciate that. And without further ado, Ben McDonald, take it away.
Fantastic. Thanks so much, Beatriz, for the welcome. And I guess just being you, you've made my time here at MIT so wonderful. And it's a real pleasure, I guess, to have this opportunity to reflect on how I got here and take a bigger picture view. So the talk I gave last year was more focused on my work, I guess, currently in the Swager Group. And so this will be, just a broader view of where I've come from and where I'm going. And I think before I dive into the talk itself, I wanted to talk a bit about who I am, and again, I guess how that informs where we're going. And so I actually grew up in a small town in Ohio called Ostrander, and I was an interesting kid, being the child of a plant physiologist and a scientist in the U.S. State Forest Service, and as well as my father, who was a science educator. I would say that science is very much in my blood, and it's pervasive, I guess, in my persona. And this kind of persona is further, I would say, formed by this relationship with the natural world through both, I guess, my mom's career, but additionally from my family's roots, which is actually in my father's family's roots, which in the Triangle of North Carolina, where Actually, he grew up as a tobacco farmer and a sharecropper, and that relationship with the land and our physical environment, and being the first generation actually to grow up as something other than a subsistence farmer or a sharecropper or a slave, has actually really informed, I guess, my perspective. And so the first science project I ever did was actually, I think this was from my 7th grade, where I took some of the manure from my animals and some animals of friends, and I actually looked at how different animals underwent different animals' manure underwent anaerobic fermentation. And I got to characterize that using GCMS at a local small university. So I've loved chemistry for a long time. Let's see. Oh, I think my slides are stuck. Here we go. Yeah, and so I've kind of undergone, to get to where we are now, I've undergone a long journey doing my undergraduate studies at UNC Asheville, my PhD in organic chemistry at Northwestern, and then journeying here and soon onwards. And finally, I think what's been additionally pervasive in this person that has been me is this question of why, and as it pertains to many of those images that I showed you, and understanding our physical world and physical reality. And so Fernanda was interested in the Swagger group and myself as an MLK scholar, and took the time to interview me, and I thought it was funny in that maybe I wasn't a frustrating person to interview, because I'm a person that answers questions with more questions. But anyways, getting down to why am I a chemist, I suppose it's because I found it to be kind of the central science, as it's noted in this famous textbook, and additionally, more recently, this new ACS journal, and as it gives us a molecular basis to understand so many of these phenomena that we find ourselves surrounded by, right? And additionally, we find great importance in general as an industrialized society in chemistry, right, as it underpins medicine, right, the cultivation of foods, the development of materials, right, for housing, and additionally how we communicate. But personally, I found that it's a series of models that again give me the power to understand fundamental questions of interest to myself. And as a younger student, what I found particularly fascinating was this first description, and what's kind of considered the birth of organic chemistry, and oftentimes purported to be the end of vitalism. But that's the synthesis of urea by Frederick Wöhler in the early 19th century, wherein he took what were considered to be inorganic or non-living materials, and created a material that is associated with life. Kind of one of the contributions to chemistry that ended this notion that there is a life force consistent with organic entities or compounds that is not existent in inorganic. So really moving what we consider the line between living and non-living entities. And then more recently, there's just so many interesting questions that chemists are additionally answering, such as really, for example, Juan Perez Merceder asking what is the nature of life? And so creating these polymer vesicles that are similar to living cells that when provided nutrients or monomers, and then provided energy through light, undergo these divisions, these cycles of growth, collapse, and growth and collapse, not unlike living entities. And then additionally, asking these questions of what is the nature of consciousness, and this state that is very hard, and someone with much more expertise than I could give many hours of the lecture on this potentially emergent property that arises from life. And I'm probing this using molecules found in nature as well as synthetic molecules. But what I would say is that what interests me, and as a person of from that background being a doer and maker of things. I'm particularly interested in synthetic organic chemistry and this vast space that could characterize it. And I'll refer to this as organochemical space. And so one of my favorite books as a kid has a quote that I think kind of sums up the nature of organochemical space, and that is just mind-bogglingly expansive. And even just considering small molecules less than 500 Daltons, there's essentially, in mind-bogglingly large, again, a set of compounds that can exist. And I'm just kind of putting up some molecules here to show you the structural diversity that can exist within even this constricted space. And also kind of showing some of the nature of, I guess, chemistry that I found most intriguing with respect to organic chemistry, and that is the fact that, again, there's so much information contained in even these small structures. As an undergraduate student, I've found these to be essentially hieroglyphs that contained more and more meaning as the more you learned about chemistry itself. And then in the Swagger group, my attention has been turned to macromolecules and the abundant space that can exist within there, with one example being the fact that a simple 100 mer or a polymer of polystyrene that contains 100 units contains more possible stereoconfigurations, that is the spatial relationships between these stereocenters, these chiral centers. Here, there are more iterations within that very short chain than there are even essentially unique molecules, and there are grains of sand on the earth. And so while being, I guess, captivated by this, I've also been captivated with the way that humans have sought to explore the space. And that's always through the natural world as that guide, and particularly an inspiration in terms of how we design reactive chemical systems, and particularly the reaction that got me hooked on organic chemistry and wanting to, again, be a maker as well as a studier of these compounds, was the way that this simple a linear compound squalene is cyclized into, in this case, either hopping in prokaryotes, or if you don't close this ring, into cholesterol. And just the sheer amount of precision that exists, and that is in a world that we can't see with our own eyes. And more recently, on the macromolecular front, thinking about how nature takes very simple molecules, such as glucose, the sugar, and strings it into polymers that are hierarchically structured into fibrils that are then further aligned in three-dimensional space to give us some of the remarkable properties of wood, and that being having very interesting tensile properties, as well as the ability to have this moisture-triggered actuation. Excuse me. And so the unifying theme, as I suppose, as a human being interested in thinking about how I can manipulate molecules, is that there's this accompanying essential theme among all of this is controlling the position, the position of a chemical system, and guiding it along an energy surface. And so this just being kind of an example of one such energy surface, you can imagine as we move to, say, the redox states or different shapes of molecules, you can imagine those having an accompanying energy. And of course, nature has spent millions of years essentially carving out the lowest energy and exploring and carving out the lowest energy pathways along points in the space. And so myself, as a synthetic chemist making small molecules, doing catalysis, or making responsive materials, some of the things we'll talk about today, I'm just a manifestation, I suppose, of nature's ability to kind of move things along this, and I can be, I guess, further inspired moving forwards. And so what I wanted to discuss today is some of my work as a PhD student towards the end, where I thought about reworking Lewis-based catalyzed reactions, or reactions that that actually an enzyme cofactor does, and additionally designing responsive macromolecular systems, particularly say something that is akin to our skin, that autonomously or responds on its own to chemical warfare agents and is able to be regenerated. And so we can effectively gate evaporative cooling. So starting with the story of reworking N-heterocyclic carbenes. And I did this work with Professor Carl Scheidt at Northwestern. This area of what's known as organocatalysis kind of sprung out of the action of thiamine, which is vitamin B1, which is a cofactor in a number of enzymes, one of them being pyruvate decarboxylase, and it acts as a Lewis base to actually take these 2-carbon pyruvic acid fragments that you get from glycolysis or the metabolism of sugar, remove one of these carbons as carbon dioxide, and then undergo oxidation of this intermediate to yield acetyl-CoA, which can then go into the citric acid cycle, right? Our metabolism cycle. And from this has, again, this kind of arisen this whole field of organocatalysis that has, rather than been interested in, I guess what we'd say, metabolism, or the breaking down of these carbon-based structures, to almost catabolism, or building them, and harnessing this kind of reactivity found in nature to come up with new, what we'd call reverse polarity mechanisms, wherein we take things that would typically accept electrons and we turn them into nucleophiles. So in the case of typically of aldehydes, right, we turn this carbonyl position, we can turn this carbonyl position into a nucleophile, right, that is able to undergo reactions with electrophiles or otherwise oxidatively. Or additionally, we can take what would formerly be an electron acceptor in this conjugate position and turn it also into a nucleophilic species. And this has arisen from the seminal work by the Japanese chemist Yukai, who performed these condensation reactions with these aldehydes, first with cyanide, and then ultimately Ron Breslow realizing that the action of this vitamin B might actually undergo the same types of chemistry. And then a whole field of asymmetric catalysis, the formation of chiral molecules, has arisen from this. And what particularly what I like to highlight, one of the best applications of this is the synthesis of iridoids. In one notable case that would be of interest to us is actually taking the substrate of the iridoid synthase and in a single step being able to turn it into, in this case here, racemic naphthalactone, which is a cat attractant that my cats happen to like quite a bit. And additionally, it can be used to make an enantioenriched petallactone. And so this is additionally within nature is a kind of a central intermediate that is pervasive in many species of plants, and ultimately is elaborated into metabolites that have a wealth of biological activity in humans and elsewhere. But what I was interested in my PhD was, how can we move beyond this 2-electron manifold? Because there were a number of limitations, typically within substrate, and can we get to new bond forming and reactions? Particularly, can we mimic this kind of oxidative chemistry here, with this being what's termed the Breslow intermediate. And what one might seek to do first is to mimic, right, the action of nature, wherein you have this oxidation-based chemistry to yield new useful intermediates. And so what if we remove simply a single electron we could get to this keto radical species, which are known to be quite stabilized. And we even have this aromatic structure here that might stabilize it. And then we could undergo a further stabilize it. And then we could undergo a radical coupling reaction that would then bring us to back onto the typical N-heterocyclic carbene catalytic manifold, wherein you have this tetryl intermediate that collapses and yields you a product, in this case, this oxidized product. But interestingly, in surveying the literature, this kind of approach had been considered, but the result was actually that there is uncontrolled oxidation, as in it's actually easier to oxidize this keto radical than it is easy to oxidize the Breslau intermediate. So we kind of slip down this energy surface uncontrollably, and we get turnover of what's called this acetylcholine with other nucleophiles to always yield us these oxidized ester products. But we're interested in making carbon-carbon bonds. And so the insight that I had was fairly straightforward, and that was, what if we just did it backwards? And so if rather than trying to kind of divert the pathway down into potentially this local minima, what if we pushed our way back up it? And the way that you can imagine inputting energy to potentially go uphill would be through the use of light, right? And so integrating what has now become a broad area in synthetic organic chemistry, and that being photoredox, wherein we use, say, a photocatalyst that upon photoexcitation is able to undergo these electron shuttling events. And so one way that we might do this is to have a species that upon interaction with the photoexcited catalyst might donate an electron and then to generate a radical, as I've shown here. So in this case, this is this Hansch ester derivative that upon oxidation undergoes this very quick decomposition into this pyridinium and this stable benzoic radical species. And then we can take that electron that we had removed from this substrate and put it into our acetylcholine, actually then turning it into this keto radical that could then undergo this radical cross coupling and then decomposition of this intermediate, yielding us the desired product. And so it was very gratifying to kind of pursue this line of thinking with a younger PhD student who took this idea and developed actually a whole new strategy for the high throughput screening of these kind of pathways of interest, and you can read here in this PNAS article, so that we could go very, very quickly right from this idea that I had scratched out into an actual realized chemical system. And you can see, at least in this case, we use a little bit of a Lewis acid to facilitate the transfer of the electron to this keto radical. And ideally, and more recently, we've developed this or elaborated this into a catalytic variant, wherein we can actually take a carboxylic acid and turn it in, activate it into a species with a good leaving group, and then use the NHC catalytic pathways as we had desired to undergo these cross-coupling reactions. And so this has kind of become a burgeoning area of dual catalysis with others, such as the preeminent radical chemist Armido Studer, recently providing a wealth of beautiful examples. And so while I found the kind of the navigation and I guess of these energy landscapes to be pretty interesting, I also had, I guess, a broader set of interests that went beyond just turning, I guess, one molecule into another. And I wanted to, again, simply make things that did something that was useful to us. And fortunately, Tim has provided me the opportunity to do that, and I've got to work on engineering a basically a set of composite materials that, as I kind of alluded to in the beginning, act as a second skin and respond to chemical warfare agents to provide an on-demand protective barrier. And we'll get to the relevance of that. But what I wanted to highlight was the fact that our skin is actually additionally an organ that acts as a selectively porous barrier, right, that protects us from microbes and more recently external threats such as viruses, but is also able to respond and cool us, the greater, I guess, organochemical system, if you will. But unfortunately, there are other agents, particularly chemical agents, that can be even designed to penetrate this barrier. And so it would be of great interest if we could develop, say, an additional layer, a second skin that could still enable this evaporative cooling, but then upon triggering by this chemical agent, collapse and protect us, and then be able to be, again, regenerated. And phosphorus chemical warfare agents are one such chemical entity that have been around for a period of time and been undergoing development for essentially the entire 20th century. And so starting what are known as the G series, such as sarin and some other ones, Soman and Tabun, these are still very toxic. They are, particularly sarin being around 80 times more toxic than cyanide, still quite a bit more toxic than phosgene, these being chemicals that other synthetic chemists probably would have handled. And additionally, again, more toxic than sulfur mustard, which we'll come to. But these have undergone further development into materials that, rather than being vapors, you can actually be absorbed through your skin, and with an estimated LD50 of around just several milligrams. And then more recently, there have been a new series of these agents developed known as Novichok agents that are, again, still almost an order of magnitude more potent than VX itself, but additionally can be kept as binary mixtures and, say, disguised as simple pesticides that upon mixture actually mixing in the field realize this extremely potent agent. And the way that all of these compounds work is that they essentially disrupt our ability to breathe. And so they prevent the degradation of this neurotransmitter acetylcholine that typically acts across the synapse, right, to stimulate the firing of a muscle, and then this esterase comes along and basically clips off this acetyl group, and which renders it inactive and allows the muscle to relax. And so again, this buildup of acetylcholine effectively makes nerve impulses unable to be reset. And so it's sometimes said that these compounds actually inhibit your muscles, the muscle systems that are associated with breathing, but actually some of these are so potent and are so active at such low threshold amounts because they actually act further up the nervous system and inhibit some of the aspects of your nervous system higher up in your respiratory chain. And they actually just prevent the nerve impulses being sent to your respiratory system to tell you to breathe. And so in the world where we find these agents, well, if you're a 90s kid like I was, you would remember it from the blockbuster hit The Rock, featuring Sean Connery and Nicolas Cage, where they are saving San Francisco from a rogue general from sending missiles containing these compounds into the city. But if you were an adult in the 90s, you would remember some of the horrific events, particularly one being an attack on the Tokyo subway during rush hour, where somehow only 13 people were killed, but thousands upon thousands of people were exposed to these compounds and were injured. And more recently, these are actually been deployed as tools of assassination with Kim Jong-nam, I believe the half-brother of Kim Jong-un, being assassinated in a Malaysian airport, with VX, a um, Russian spy and his daughter actually being attacked in the UK, with the Novichok agent, and then additionally individuals who actually found some of the discarded the apparatus that was used in the attack, which was just a perfume bottle, were also sick, and with one person dying. And then most recently, the Russian opposition leader, Alexei Navalny, being attacked with a new unknown Novichok agent in Tomsk before a flight in Russia last year. And so these are still very pressing, I guess, threats to the safety of civilians as well as military personnel worldwide. And we're interested in developing the next generation of personal protective equipment that, again, are not necessarily interested in sequestering the materials, but actually responding to them, destroying them, and protecting you. And so the way that we've gone about this in developing the second skin is actually growing polymers from a surface that has carbon nanotubes aligned in a polymer matrix. And so these carbon nanotubes actually are very effective water carriers of water moisture vapor, as well as excuse me, condensate, where due to the tiny diameter of these tubes, the water just tumbles through very, very quickly. And we have developed polymers that actually repurpose this dye that reacts with these electrophilic agents to then become ionized. And they actually, it's interesting, they undergo what I would call the contrary or contra electrolyte response. So typically when something becomes charged, right, they show their, and so on polymers, when you have those charges stuck to a polymer chain, that just makes the chain elongate. However, these are able to undergo pi-pi interactions and are actually attracted to one another. And so this results in these polymeric materials going from this sterically, this kind of repulsion state where they're all pushing away from each other and our chain extended, to actually this collapse state. And so we're particularly interested in developing a response to these G agents initially, but thankfully I get to simply use these simulants in some of these studies. And so I'd like to share some of the more recent work wherein we've been interested in developing, applying this scheme to stretchable materials, right, ones that you might expect to find in clothing garments, and additionally expanding this response to another agent, these mustard blister agents. And so with kind of these two key requirements in mind, we've been developing a generalized functionalization strategy that lets us, work on a number of different surfaces, but then also at the same time investigate building in pieces to respond to these agents. And so the way that we have generalized this scheme towards robustness, right, and again, the versatility, as I said, was to, rather than simply growing the polymers directly from the surface, we actually will use an interstitial polymer layer that we can attach to the surface, but that doesn't exhaustively react to the surface, so that we have these reactive anchors that we can use to either do what I would call a layer by layer, or really this is a covalent layer by layer reactive process, where we have, say, a polymer that has reactive nucleophilic amines that will react with this polymer on the surface, and then we can simply iteratively subject the surface to this amine polymer that right, does not fully react and leaves these reactive amines, and then we can add in, right, solubilized Ti polymer, leading us back to this sort of surface. And there are some benefits to that. We can know the exact size and composition of these materials as we put them on the surface, or we can use this Ti polymer more as a amplification modality, wherein we, rather than right, the surface, the number of grafted polymers grown from the surface being dependent on right, the number of linkers on the surface, we can decouple that and put on different tie polymers. And this allows us to do this a much more rapid and expedient functionalization process. And it gives us this kind of topology that more similarly mimics our previous strategy, which we had shown to work. And so we have, you know, I've kind of demonstrated a proof of concept for both of these. It just happens, not going to really go into details today, but it just so happens that this strategy, again, probably due to the topology of or the way that the polymers are oriented with respect to each other, that being anchored and aligned, happens to respond and show that similar collapse. But more interestingly and more on topic with thinking about designing reactive organic systems inspired by nature, I'd like to talk a bit about expanding this reactivity to sulfur mustards. And so there is actually a key difference between these two chemical warfare agents and their chemistry and I suppose their mechanism of action. And that being that these phosphorus agents are what I would characterize as persistent electrophiles, as in this, the chemical species that you would be exposed to is the reactive species. And so, right, this will react directly with whatever constituents. In this case, we've just drawn the responsive unit to say be activated and turn into a good leaving group and yield us this phosphate in this triaryl cation. But in the case of the sulfur mustard, actually the reactive intermediate is this is a thiouranium and not this alkyl species. And so really the reactive species is actually in equilibrium, and the environment, if whether it be a solvent or whether it be the surface, is really determining the position on this equilibrium. And this is all to say that if we wanted to enhance response or create a response, because it doesn't exist with our current materials to this agent, we need to think about, rather than just dialing up the reactivity of the surface, we might need to actually think about adjusting the composition of the surface to actually enhance the reactivity of the agent itself. And thinking about, making something that you would, be economically feasible to deploy in a suit, particularly being, say, a standard issue suit. So making a membrane that would be incorporated into a standard military uniform or a first responder uniform, rather than it being this separate suit that you have to suddenly put on once a threat has been detected. We need to have this long-term stability. We need to have reusability, and then there are some some of these mechanistic character pieces that I had just kind of gone through. And particularly, how are we going to get this into our polymeric materials, as these the units that we might seek to incorporate are not compatible with the chemistry that we're doing, say that ROMP ruthenium-based catalysis from the surface. And so what we settled on, and in terms of thinking about long-term stability and the economy and availability of such entities, we could think of repurposing some of the reactive elements, say, found in the catalytic triad of an esterase, and notably the actual substrate that the phosphorus agent actually goes after. And so thinking about incorporating these Lewis base or Bronsted base entities as well as nucleophiles. So thinking of cysteine as an amplified version, or a more reactive version of serine, and then again potentially taurine and aminoglycerol as those entities to modify the polarity of the surface and, say, enhance again the generation of that thiouranium reactive species. And the way that we would incorporate these entities is through what we would call a post-polymerization modification strategy, that being that, again, we use our handy amine reactive ester functionality so that we can actually make, say, a single surface and then functionalize it with, say, one of these multiplexers or a variety of them, and then think about characterizing on their response. And so as a first pass, what we did is actually replace one of our hydrogel units on the surface, this PEG diester, with this pentafluorophenol ester, as we are going to be using some particularly hydrophilic groups as multiplexers. And the way that we go about characterizing these materials, and that would be actually quite convenient, is that we can use, again, this dye as a colorimetric indicator of response. And then we can use this, if we use, say, glass slides that are particularly smooth, we can actually etch away some of that polymeric material and do some, and I'll show later, some profilometry to understand the thickness of the polymer layer, and again, to characterize that change in polymer configuration that we'd like. And so the way that this kind of crude vapor generator system that I had built, that allows us additionally to control the humidity, as this is, again, a very pertinent and important factor in the ionization of the mustard agent. We can basically run different ratios of a dry air and a wet air over a chemical warfare agent that then will be volatilized, and then we can have in a separate chamber our functionalized glass slides, in this case, that are representative of this grafted polymer layer, and we can assess their response over time. So I'll just kind of show you, this is some recent data. Thinking about the response of our grafted materials to the sarin stimulant, which is DCP, you can see with a 30-second exposure and without a multiplexer, we only have a very a mild response, as evidenced by color. However, when our multiplex materials, where we've, say, replaced or we've incorporated, say, cysteine, this emitted alkylamidazole, we see a greater response. And then additionally, some of these other mixtures, we can see an increasingly strong response, notably with the aminoglycerol combination with the cysteine ethyl ester, and notably taurine, which we're more interested in for the purpose of the mustard, still actually ends up enhancing the response to DCP, which could be for a number of reasons. And then what we're actually quite interested in is enhancing this response to our mustard agent, which in this case, the simulant is what we call half mustard. So is actually less reactive than the mustard agent itself, and there would typically be another accompanying chloride on here. And then after, say, a 6-minute exposure in these conditions, we can see there's virtually not much of a response at this lower humidity. However, we can see that we do get a greatly enhanced response over the course of, say, 5 minutes, up to half an hour. We get a very strong response. And the final remaining question, well, this is just a nice proof of concept, and that will need some more follow-up, but we need to figure out, are we still getting collapse? Because we've really disrupted this system. And so what we do is we take our glass substrate that has these grafted polymers, and we put on what's called a shadow mask that basically is a grid that has apertures that allow plasma to pass through, and it leaves us a pattern surface that we can basically run a tip over, and that will tell us with either with an AFM or either a profilometer, that will tell us the height of these grooves. And so so here we can see in our standard composition, when we expose it to TFA or trifluoroacetic acid, our simulant, we do get a nice strong collapse. However, unfortunately, in these other cases, we're now getting the standard polyelectrolyte response in this case. However, in these other cases where we've added in an additionally charged species and a charged species, an anion in taurine that should be complementary, we're expecting to see a tighter collapse, but we're seeing a collapse that's seen in what are called polyampholytes, where you have basically these complementary charges on a polymer that usually stick together. We're not seeing that, so we need to investigate that further. And that might actually just be to the fact that we're doing this all in the vapor phase, because the mobile counter ions are not being necessarily washed away. So may not actually have a polyimphelate response, or the typified polyimphelate response that you see in solution may not be expected. And these are useful for if we had a microporous material where we have the polymers inside of pores and they were opening and closing. But for our nanoporous materials that will be grafted on top, we need to get response. And so we're following up on that. And so this has been a pretty, I would say, it's been truly a joy to kind of take the ways that we might think about designing catalytic small molecule reactions that happen in solution and then engineering them to happen, say, attached to a polymer on a surface. And lately, the last few months, I've spent considering, or I guess pulling my head off, figuring out where do we go, where do I go from here? And so what I hope I've showed you today is that I've learned quite a bit from repurposing, nature's reactive motifs and pathways, but how do I establish, I would say, my identity moving forwards as an aspiring academic? And, I spent some time thinking about this, some time reading and writing, and what I've realized is that I'm still very interested in, well, I've done, I guess, what I would say is fundamental research. I'm quite interested in what is typically called, and this is past year's research quadrant, use-inspired research. So either applied applications driving fundamental research, or fundamental research driving things oriented towards an application. What I've realized could be the possible synthesis of these activities is moving on from the biomimesis of small molecule reactivity, right, on whether, again, be on a surface, whether it be in solution, and thinking more about how this reactivity is carried out in space and time in the assembly of biological materials. And so I was quite happy to come to this realization. So just to chat a little bit more about biomimesis and material systems, I would say that, right, the form of nature has consistently captivated, right, I guess, right, human imagination, as most typically, I guess, demonstrated in right Da Vinci's design of a human flying apparatus, but it's still carried out even now. Right, there's a striking similarity between some of our military airplanes and birds, and maybe something that is more relevant to a chemist and my interest on the micro to nano scale, thinking about mimicking the function of some of the structures of these very small structures found in nature. One good example being that dry adhesives of the gecko feet that use dispersive interactions with substrates that arise from the formation of these very small structures on the surface of the pads. And this has been mimicked in nanofabrication modalities. But what struck me is that we oftentimes still use it's still this top-down engineering paradigm wherein you take a material and its properties and you either build it up, right, and kind of copy and paste it into the bigger structure that you would like, or in this case, right, we pare down those features. And so we're not really getting, I guess, what I would say those emergent properties that are commensurate with living systems. And of interest, right, again, to something that I just talked about, like carbon nanotubes, there was this great fascination, you may recall, in the once we realized the extraordinary tensile properties of carbon nanotubes, it was thought, wow, if we can just take the strength of an individual nanotube and weave them together and, again, build that out into a large macroscopic structure, we should be able to simply lift things from the ground into space. And we might say repurpose, right, a millennia-old technology of weaving or fiber formation towards this. But unfortunately, what this process kind of ignored is how defect sites arise in this. And so if you don't necessarily link the ends of the fibers together. And so the response to this was that, well, we just need to grow multi-meter long carbon nanotubes, because then by making them continuous on that length regime, we would reduce the rate of defects in this fiber, such that we should realize those tensile strengths that we want. So while we can grow very long carbon nanotube, very long carbon nanotubes, I don't think anyone's done multi-meter long carbon nanotubes yet. And so we're still waiting for that, but a way that nature could inspire us to solve some of these problems are thinking about the structural hierarchy and the chemistry that occurs on fibular biomaterials, like those of collagen, right? And so collagen is this, I've represented it as this little charged cylinder here, but it's a helical tripeptide that, right, has these charged ends, right? Again, you have it, that being your N terminus and C terminus, that are spatially confined and behave as essentially liquid crystals, and all align in our enzymatically cross-linked, and then into fibers that are then further structured into materials such as our tendons and our connective tissue. And again, versus the individual properties of tropocollagen, we have new, very strong elastic or very strong stiff materials that can arise, and all of this through the spatial orientation of these materials. Another facet here being particularly captivating is, and as I talked about before, was how cellulose is essentially, from a polymer chemist point of view, a surface confined polymerization-induced self-assembly. So you take soluble sugar and you polymerize it across a membrane into polymers that are no longer soluble in water, and you do this with hierarchically grouped polymerases that then you start concurrently extruding polymers that snap together in groups of 3, and those groups of 3 are typically grouped another 6 to get these typically 8-chain fibrils that have these crystalline and amorphous regions that allow some of the mechanical properties that are seen on these materials, and are further structured into macrofibers that even are helically aligned as you look down the structure of wood. And
Ben? Hi, we have about 3 minutes left. I didn't want to interrupt you, but I also wanted to be mindful of yours and everyone's time here.
Mm-hmm. Oh yeah, I think I have 2 slides left.
Oh, perfect.
I almost timed it perfectly. Thanks, Beatrice. Yeah, so what I would say is that in thinking about, I guess, these interests as a chemist and working chemists contribute to some of these unmet needs, and being the spatial chemical tailoring on assembled materials, I think there are many great opportunities there. And so I'm happy to say that we'll be able to pursue some of these questions in nanostructured organic materials through the context of polymer chemistry at Brown. And I'm very excited to be starting there this fall and to do some of this structure-based and reactivity-based inquiry to hopefully generate these new chemical tools that will let us build materials from the bottom up the way that nature does. And I'm particularly excited to kind of work in these areas that I've already had some experience in, and get to learn new things in the area of assembly, and think about what organic chemists have to contribute. And particularly excited to join the robust community of scholars and a very large presence of non-majority scholars that are arranged in this group called STEM Jazz there, led by MIT alum Chris Rose, who's an associate provost at Brown. And with that, I think I should thank a lot of the people that I have worked with along this journey at Northwestern. As I told the story of my work with Carl and Allison. And then on the second skin materials, here, Yvonne and Qilin and others such as Lennon and Sun Jin, who have helped with the characterization of some of these materials. And without this collaborative effort, none of this would be possible. And some of the work that I didn't talk about, but my collaborators, Cassie, Kong Hee, and Hari. I don't think he's in this picture, but and then of course a more recent picture here of the group with Tim and of course Brian, who, as I think everyone who's affiliated in any way with the chemistry department knows of his importance, and also Emily, who's not in this picture, and some of my other collaborators. And then of course it's been truly a pleasure to be a part of this community, particularly the Visiting Scholars Program for 2 years now. I'm getting to meet a number of the wonderful scholars. And with that, I'll wrap it up and answer any questions that anyone would have.
Can you hear me?
Yes, we can.
Okay, good. So first of all, I'm biased because I'm a McDonald. I happen to know who Ben is. And also what's really great, because a question I ask a lot of people and myself every January is, what problem are you out to solve in the world? So as I sit here and I listen to Ben talk about this second skin, it's amazing because it's something, of course, I never thought of. And I'm sitting here going, oh my goodness, this is really cutting edge. This is going to happen. So I said, congratulations to Ben, and thank you for having him be here today.